for interval or ordinal dependent covariates. What if the outcome that we want to analyze is a yes or no outcome, a binary outcome? Okay, so either the patient is dead or not dead, or the patient is discharged or staying in the hospital within 30 days, or is the patient alive within 30 days after admission. So it's an outcome that have only two possibilities, yes or no. To examine factors that associate or predict such type of binary outcome, the analysis is called logistic regression. So do age over 65, gender, race, or serum lactate level of higher than 4 predict which patient will be discharged alive within 30 days after admission for sepsis? Now, with this kind of analysis, you can evaluate what kind of categories of the patients or what kind of characteristics can predict a certain outcome. In animal experiments, very often we do experiments, especially in cancer research, we do what we call a xenograft experiments in nut mice. Have you all seen a nut mouse? No? Nobody seen a nut mouse? A hairless mouse. A mouse with no hair. Okay. Well, I'll tell you, there are mice that are mutants. These mutant mice have no hair, and they have a defective immune system. And therefore, they do not reject cells that are transplanted into them. So we use these mice to test cancer. We inject human cancer cells into these nut mice, and the nut mice is not able to reject the human cell. And these human cells would grow tumors, and the cancer can grow and sometimes spread. And because the human cells are grafted into these mice, it's called graft, okay? Xeno. Xeno means foreign, not the same, okay? Meaning that you're grafting cells of a different species. Okay? You're not grafting mouse cells into these nut mice. Okay? If you're grafting mouse cancer cells, it's called allograft. Okay? So here we're grafting human cancer cells, so it's called xenografts. Okay? So one easy type of experiment is to inject the human cancer cells under the skin, on the back of the nut mouse. Why we pick the back? Because the mouse can scratch on it. If it's on the back, the mouse would not be able to scratch on it. So after you inject the human cancer cells, you inject equal number of human cancer cells into a group of mice, and each mouse would grow a tumor. And how fast does that tumor grow? There's individual variability depending on the mouse. So you measure the size of the tumor on each mouse over time. And when you test the effectiveness of a drug, you inject the drug into the mouse. You divide the mice into different groups. For example, here in this experiment, I divided the mice into 3 groups. One group is the control mice. that received injection of placebo, the saline, no effective drug. The other second group received this drug called AZD1152 at this dose of 62.5 milligram per kilogram per day. And then the third group received this same drug, AZD1152, but at double dose. So now I have 3 groups of animals, and we'll follow the size of the tumors over time. Now, how do I analyze the growth curve of these tumors and test and evaluate whether the drug is doing anything in suppressing the tumor growth? What statistical test can I use? Okay. So if at the end of the experiment, I cut the tumors out and weigh the tumors, I have the tumor weight and down to the precision of milligrams. Then I compare the weight of the different tumors among the groups. Okay, now here I have 1 group, 2 group, and 3 groups. And I have the mean tumor weight from each group. Is it appropriate to compare between groups using the t-test? No. First of all, there are more than 2 groups. T-test is not appropriate. Then I should use one-way analysis of variance. The next question is the distribution normal? I don't know yet. They have to do the normality test. If the normality test fails, then it will not be the regular one-way ANOVA. It will be the Kruskal-Wallis one-way ANOVA, Kruskal-Wallis ANOVA, the non-parametric ANOVA. So one step by step, you need to follow the first slide from this session that I shown. There are 4 questions. How many groups are there? Is it a continuous variable? Are they independent? Is the distribution normal? So you answer these questions in order to pick the right test. But then, look at here. If you analyze the data or analyze the tumor size at the end of the experiment, you are looking at one time point at the end of the experiment. What about all this data that happened before? Do we use it or do we not use it? So in order to use the data that are collected during the course of the experiment, okay, the data over time, there's a method called growth curve analysis. So you average the growth within the population and its variation using linear model. So if you have these data points for different days about the tumor size over time, from that, the data from each animal, you can construct a linear growth curve, and a line with intercept and the slope. And then you analyze the what we call latent variables, the intercepts and slopes, and analyze these among the groups. And this type of analysis, the linear growth modeling, would be more powerful because it takes into consideration of all the data before the end of the experiment. And again, I would like to go back and emphasize the graphical presentation of data. First thing, color. Use color for a purpose. Don't use color to distract. For example, here, you use the color to represent different groups so that it is clear that there's clear separation in the distribution of the body weight between these two genotypes. Contrast. You make the lines heavy and thick. Okay. Colors, if you use colors with big contrast so that you can see clearly from far away. The bottom line is from far away, as well as you should be able to see it clearly when it's shrunk down to a significant, much smaller size. The lines need to be thick, symbols need to be big, fonts need to be large. The size. Okay. When you initially construct the graph, you need to do it in the large size so that when you shrink it, these days people use the electronic media, okay? So that when you have a high resolution, when you're shrinking it down to a 600 dot per inch graphical file. Okay. If you do it large, so that when you shrink it, you have good resolution. If you start out with low resolution, you may end up going back to do the graph again. It's always better to do it right and do it once and save time. Common types of statistical flaws. Very often is in the study design from the very beginning. When you do your experiment, when you design your study, you need to have the statistics in mind. If you don't plan ahead of time to use what kind of statistical test or to evaluate what kind of variable or what kind of data you're going to collect, you're going to be in trouble. And also you need to be careful in design